It is important to remember that there are diseases out there like sickle cell anemia that result from one DNA letter change in the gene that codes for a different amino acid at a critical region of the protein, which makes the red blood cells of the patient go from healthy red blood cells to sickle cells, as shown in these photographs. So this is to emphasize that one DNA letter change out of 3 billion can cause a serious genetic disease. This is fluorescence in situ hybridization, and this is technology also used by cytogenetics to look for missing or extra pieces of DNA. Another technology that has become very popular in genetics in the last decade is microarrays. Microarrays are much like a karyotype in that they tell you if the patient's chromosomes have missing or extra chunks of DNA. Microarrays were being used in the late 1980s and early 1990s to look at cancer cells to better understand cancer in patients. Cancer cells typically have a lot of mutations in missing or extra pieces of DNA. Microarrays are also called gene chips. Basically, you take cells from a patient and you open the cells to isolate the DNA, and then you chop the DNA up into smaller pieces, and then you label the DNA with different dyes, and then pull the strands apart to make single-stranded molecules. You put the single-stranded molecules of DNA on a special glass slide. The slide has millions of single-stranded DNA segments on it called probes, which are glued to the slide. The patient's DNA has been tagged as well as DNA from a reference control that we know is normal. These are both added to the slide with thousands of different probes. The slide is then put into a magic box, which is basically a machine, with a special laser inside, which scans the slide and tells you what ratio of binding you have of the patient DNA and the control DNA that are attached to the probes on the slide. So this is a technology to tell you whether the patient has missing or extra pieces of DNA based on the ratio to the normal control. This is an example of what a microarray machine and computer would look like. So the front of the box would open up, and then you'd be able to put the slides into the machine, and then the laser is inside. So there are thousands of probes used for all different chromosomes. All this information is analyzed by computer software, and then you get a printout which looks like this. Actually, this is my chromosome number 1 and number 18. So 20 years ago, microarrays were used to study cancer cells. The number of probes that were used were pretty small back then compared to what we have today. At about 2005, there were microarray tests available which used 44,000 probes to look at all the chromosomes and gene-rich areas of the entire genome. You could easily see missing or extra chunks of DNA down to the half a megabase size, which was about 5,000 nucleotides long. There are now microarray tests using over 1 million probes. Part of the problem here is that we can see smaller and smaller regions of DNA, but we do not know what some of it means. If you have a missing region of DNA that is, say, 50,000 units long, you can't say for sure if this is the cause of the patient's seizures or developmental delay, unless you can look at published studies done by other scientists who showed that this finding yielded these outcomes in other groups of patients. The technology for this type of microarray is so new that a lot of the clinical correlation studies just haven't been done yet. The word genome means all the genes needed to make an entire individual. It is the complete set of blueprints to make up the individual. Humans have about 20,500 genes in their genome. When we perform microarrays on people to cover the entire genome, we call this complete genomic hybridization arrays, abbreviated ACGH. Hybridization refers to the binding that takes place from the single-stranded patient DNA with the other stranded DNA glued to the slide. We remember that to make a human you need a sperm and an egg. The sperm has 23 chromosomes and the egg has 23 chromosomes. The first 22 chromosomes are called the autosomes, and the last chromosome is called the sex chromosome. The sperm carries either an X or a Y for this last chromosome, and the egg always carries an X. When we have a sperm and an egg joined together to form an embryo, we have an XX, which makes a girl, and when we have a Y sperm and an X egg together, we have an XY, which is a male. So note here that the father's sperm always determines the sex of the child. So now we can talk about genetics a little bit. A genome is a complete set of genetic instructions for an animal. All the genes needed to make an individual. We have 20,500 genes in our genome. The sperm has one complete set of genes, and the egg has a complete set of genes. So the point here is that we need 2 copies of every gene to make for a genome. In genetics, we talk about Mendelian disorders. This term is from a monk who lived about 150 years ago, who worked with pea plants to get yellow pea plants or green pea plants only. His name was Gregor Mendel. Here's a painting of him. This led to an understanding to a lot of things in genetics, including autosomal dominant and autosomal recessive. So when we talk about Mendelian genetics like sickle cell anemia or cystic fibrosis, we are talking about single gene disorders where a mutation in one gene causes a disease. There are over 6,000 single gene disorders identified, and many of them are quite rare. We'll stop here and go to the next slide.